I'm Dr. Marsha Pierce, and I'm an assistant professor in the Department of Pharmacology at Midwestern University in Downers Grove, Illinois. And I'd like to thank you for joining my talk, Preclinical Evaluation of Marine Natural Products for Neuroscience Drug Discovery. By the end of this talk, participants will be able to describe the drug development process and timeline, identify challenges associated with neuropharmacological drug development, learn about how the marine environment contributes to unique metabolite structure, and learn about upcoming marine natural products in preclinical and clinical use. So how does a drug get to market? Initial hits through screenings and synthesis identify a compound that binds to a target of interest. And when those compounds are identified, they generally don't have the appropriate properties that will get them to market initially. They're going to have issues with solubility. They're going to bind to things that you don't want to. You can have side effects. So that initial synthesis, examination, and screening occurs in various cellular cultures. And when we have identified a compound of interest, as you can see in the bottom of this, that could be anywhere between 10,000 and 25,000 compounds through this basic research screening. We then move approximately 10 to 20 of them to the preclinical testing in animals. This preclinical testing generally takes place in two different animal models, one rodent, generally mouse, and one non-rodent, generally rabbit. Now, in these models, it looks for ability to cross the blood-brain barrier. It looks for dosing, safety, side effects, target effects, what it could potentially treat. They also do longer-term studies looking for carcinogenicity, for genotoxicity or mutagens, and especially for reproductive toxicity, looking to see if the male mouse is then able to go on and reproduce or the female mouse and so forth. If the compound passes all of these initial tests, an investigational new drug application or IND application is shown at where the red arrow is located. And if that investigational new drug application is approved by the U.S. Food and Drug Administration, the FDA, it goes into your initial phase 1 clinical trials. About half of the compounds can make it into this phase 1 clinical trials looking for safety. First, we're looking for the appropriate dosing. We want to make sure that there's no major side effects, and we want to make sure that the compound is safe in humans. Next, if it manages to achieve all of these, then it moves forward into your phase 2 clinical trials, at which times we're looking for efficaciousness. Does the drug treat what we expect it to? And is it efficacious or more efficacious than current options already on the market? If it makes this goal, then we next look in the human clinical trials at the phase 3. Is it worth the risk? There are a lot of drugs that have very terrible side effects, including a number of your chemotherapeutics, but sometimes they're used and chosen because their effects on cancer are better than living with or dying with said illness. If it passes the phase 3 and is determined to be worth the risk, then a new drug application is filed with the FDA. If there's potential for abuse, that drug will also be reviewed by the Drug Enforcement Agency, the DEA. Once a drug is approved, it goes into the phase 4 or the post-marketing surveillance. At this time, they continue follow-up clinical trials and also watch for potential side effects that may have not been caught in the early phase 1, 2, and 3 trials. After 20 years, the patent expires and generics become available. Notice the attrition of these compounds over time from your initial basic research to get one drug to market. In general, it takes about 10 to 12 years and about $1 billion to get a drug to market. So, neuroscience drug development is particularly fraught with a lot of complications that other drugs don't have. For example, when you want to test a drug for, say, blood pressure, you've got a very concrete measurement of what the initial blood pressure was, what the effects are that are seen with the drug. Is the blood pressure lowered, yes or no? And then any side effects. Unfortunately, with neuroscience drug development, we're working with very complicated diseases, including Parkinson's, Alzheimer's, and stroke, with the 9 leading causes of neurodisability accounting for about $1 trillion a year in total economic burden. Moreover, there's a lot of factors that complicate this low success rate. For example, nearly all CNS therapeutics treat symptoms rather than modifying the disease. When we're treating chronic pain, we're treating the symptom of it. We're not modifying the overall effects that are causing the pain in the first place. Next, diagnoses are often based on clusters of clinical symptoms rather than objective laboratory measures. For example, a recent drug that did make it to market, aducanumab, is used for Alzheimer's disease. And it was found that this monoclonal antibody can reduce the amyloid beta plaques. Unfortunately, those plaques aren't always corresponding or correlating well with overall cognitive decline. And even though this drug has been approved, it doesn't appear to have any major benefits for cognitive effects at this time that have been identified. It also has some pretty serious side effects, including brain swelling and brain bleeds. It costs about $56,000 a year for this drug therapy to be used, and a lot of insurance companies don't pay for it. All of these combined make it a very expensive and costly process, even if it actually gets approved. Clinical trials often assess subjective measures that are fraught with both placebo and nocebo effects. Some of your drugs for your various depression or anxiety, it's looking at how the patient feels. Some of your other drugs are looking at cognitive decline, and they try to assess that in as subjective of a, or in an objective a manner as they can, but generally subjective measures are. And so you're going to have the placebo effects where people think they're getting the therapy when they're actually not, or you can have nocebo effects where people are getting the placebo but believe that they are actually being treated with said drug and therefore have complicating factors. All of these combined result in larger, longer, and more expensive clinical trials than for a variety of other medical indications. So why are we interested in marine natural products? About 70% of the world's biodiversity is found in our oceans. It can be found in fish, in crustaceans, in microbes and fungi, even in the bio-waste from the various organisms. But one of the interesting things, there are some structural and functional things that these animals and microbes use to fortify themselves, but their primary way that they communicate with one another is through chemical warfare, which, interestingly, has provided some very unique primary and secondary metabolites that we can't find anywhere else and that are not found in our synthetic chemical libraries. They have very unique structures and backbones. And a lot of that has to do with the fact that they can have high saline, the high pressure, hydrothermal pressure. You can have ones near the hydrothermal vents in the ocean. All of these microbes and other organisms have really adapted, and primarily, again, through chemical warfare. So you can have enzymes that come from that. There are fungicides, different microbes will make that can be used for treatment. I'll talk a little bit about ones that are potentially anti-parasitic coming up. Some of them make toxins. I'll talk about Prealt, which is a potent conotoxin that's used for therapeutic pain, as well as one in clinical trials, Halnuron, which is tetrodotoxin from the pufferfish. And in a way, it stops the area from signaling to the brain that you're feeling that pain. There's also a number of immune modulators. There's a number of biochemicals and nutraceuticals that have been identified that are used in cosmetics and food and other options as well. So the marine biodiversity really helps with the building of these unique chemical libraries. So now we're going to just look at some of the approved marine natural products, their derivatives or inspired drugs. And as you'll note from this slide, there's a lot of them that are used particularly for cancer. If you look at the first four in the top left corner, you'll see that those are all similar or nucleoside mimics. Cytarabine, which I'll talk about in the use of some of my studies, is one that's used for cancer because it acts as a C nucleoside, a cytosine nucleoside, and blocks then the transcription and translation of these, and then stops the ability to reproduce those cells. There are a number of other new and upcoming ones that are your antibody drug conjugates that you'll notice in the lower left corner, your brentuximab vedotin, palitzimab vedotin. A lot of these have a marine natural product that is connected by a linker, and then they'll have an ADC. The conjugate, it's going to have a monomethyl auristatin E, MMAE, or an MMAF. What that does is it finds the target cell that might express highly something that's not expressed in your normal cells. It gets taken into the cell, and then that linker cleaves, and it puts that MMAE toxin in there to specifically target cancer cells. So that's been a really interesting new way that these drugs have been developed. We also have ones, like I mentioned before, in pink in the top, second from the right. Prealt is a potent conotoxin from the cone snail that has been very effective in treating severe pain from cancer and other things. Also, the tetrodotoxin from a pufferfish halneuron is currently in phase 3 clinical trials. So there are a lot of options, even for neuroscience drug design, that occur in some of these marine natural products. So the class of marine natural products that I'm interested in right now, or the family, is called the manzamine compounds. Now, there are more than 100 manzamine compounds that have been identified from over 16 different species of sponge. And there are a number of different important factors that may contribute to these being potent neuroscience drugs. First, Peng et al. Looked at some structure-activity relationship in cell cultures, and they found that it had anti-inflammatory, it had anti-infective, and it was very important for antibiotic properties, and some of them, some antifungal properties. But one of the most important things that was really interesting that is it had some anti-malarial or anti-parasitic effects, which suggests that a single drug could both treat the infection and the neuroinflammation associated with that. Additionally, Zhang et al. Did a study using neuro-2A cells. Now, neuro-2A cells are from mus musculus, or a mouse brain that had neuroblastoma. So they're a neuroblastoma cell line, and they're a unique cell line for initial screens because they have some neuritogenic or neuron stem cell-like properties that make them useful for doing some initial screening phases. In this set of experiments, they tested manzamine compounds in these neuro-2A cells, and they saw that after 48 hours, about half of them were neuritogenic, and by 72 hours, virtually all of them showed neuritogenesis in these neuro-2A cells. Now, it's really important to note that these neuroblastoma cell lines have been known to not be as sensitive as primary neurons to various toxins. So what we're seeing in neuritogenesis for these neuroblastoma cells may not translate well to primary neurons. So that's where we then take this research by stepping into murine primary cortical cultures. So the goal of this research is to identify novel neuroactive compounds for marine natural products. We want to assess the effects on neurite outgrowth, neurotoxicity, to help identify compounds for the marine natural products preclinical pipeline. Now, all of this is done in compliance with the Midwestern University Institutional Care and Use Committee, IACUC, protocol approval, where we take pregnant gravid dams at embryonic day 16 to 18. We collect the pups, we dissect out the cortices and remove them. They are then, the cells are digested using trypsin first, and then they're triturated for mechanical separation. They are plated at 1 million cells per mil and incubated in 37 degrees Celsius at 5% CO2 with 90% humidity for 12 days. At about 48 hours post-plating, we do add the cytosine arabnoside, which I talked about earlier, the ARAC, because that blocks any of your proliferating cells and leaves us with a pure neuronal culture. Then for our initial screen, we use tetrodotoxin from the pufferfish, also known as halnuron, because it's a voltage-gated sodium channel blocker, and that causes these rhythmic calcium oscillations to stop. We also use veratridine as our positive control because that's a voltage-gated sodium channel activator from the lily plant, and that causes an influx of sodium, which then results in an influx of calcium. And so that gives us our positive control. We then screen blinded compounds using them in quintuplet on a single plate to look if there is any changes in the calcium dynamics. If we see them, we go on to do a logarithmic dose response curve with those compounds, and then also assess for changes in membrane potential using the Flipper blue membrane potential assay. So the first assay that we do at days in vitro 12 or DIV 12 is we remove the media and we apply 4 millimolar flow 8 acetyl esterase, which is a fluorescent dye, in the cell, and we incubate it for 1 hour. And then immediately prior to running the assay, we remove the fluorescent dye and replace it with LOX buffer. We place it into the Flipper 2 plate reader, which has an automatic arm that just delivers precise quantities. It also has a laser to illuminate the fluorescence and a CCD camera to capture that. In the bottom right corner, you can see that it records all 96 wells at one time, and you can see the combined result of multiple wells in the bottom right corner. So next, how do we understand the data? The data is graphed on the y-axis with fluorescence units and the x-axis over time. So for the first 2 minutes, we allow the rhythmic calcium oscillations to occur. And then, as you can see with the tetrodotoxin on the left, once that tetrodotoxin has been applied, the 1 micromolar, you see a stop in those rhythmic calcium oscillations, which occurs because the sodium channel is blocked, and that ends up downstream blocking these calcium oscillations. We're also looking for changes in the number or amplitude of oscillations, as you can see in the veratridine or our positive control. Now note that the units on the y-axis vary based on compound. So here you can see that the veratridine has the rhythmic oscillations going in the culture until it's applied, and then we see a very large sustained influx of calcium. And so that decreases the number of oscillations and has an increase in the amplitude. So we then took all of the blinded compounds. As you see, we've got tetrodotoxin in the top left corner and veratridine in the bottom right, and then compounds M1 through M21 as they were labeled. And with those, we saw a few that had slight increases in the amplitude of oscillations as marked by these green arrows. We also saw some with a slight decrease in the oscillation number or amplitude as marked by these yellow arrows. Overall, though, we did not see anything as dramatic as we see with the tetrodotoxin or the veratridine, suggesting that there's not major effects on the calcium dynamics. So in this instant, we chose not to continue with the dose response curves. Next, we're looking at neurite outgrowth assays, and we analyze these using ImageJ packaged Fiji with NeuronJ plugin. And when we're looking for total neurite outgrowth, we take an image that we have taken using a confocal microscope that has been had a cell stained with rhodamine phalloidin for the neurites and DAPI for the nucleus. And what it does is uses the pixel color to help semi-automatically trace the overall neurite outgrowth. And then it provides a readout of the outgrowth in micrometers. Now, in addition to the total neurite outgrowth, in the middle here, you see that we can look at the complexity, looking at the number of processes. In this case, there are 4 processes coming off the cell soma. And then we also can look at the total number of branches as shown on the right, where you can see that there are 15 branches total in this cartoon of a neuron. So now we're going to look at what those results look like. Here are individual cells for each compound. They were plated similarly to what I talked about in the initial calcium assays, but these are plated at 25,000 cells per well on a plate glass cover slip in a 24-well plate, and they're allowed to settle onto the glass cover slip for about 3 hours, at which time we put a single dose of each of the compounds. In this case, we chose 10 micromolar, which is a relatively strong dose, so should give us a response if we're going to see one. And from there, we looked to see if there were any increases in what would be neurite outgrowth compared to our negative control in the top left, which has just the vehicle DMSO, or our positive control in the bottom right, which has the veratridine. And as you can see here, there were a number of them highlighted in these green boxes that showed what appeared to be increased neurite outgrowth compared to the negative control. And then there were a number that showed some blebbing or other signs of neurotoxicity that we can see in the cells as highlighted in these yellow boxes. So that data is exported from ImageJ into GraphPad Prism for analysis, looking first at the total neurite outgrowth across the compounds for 10 cells for each compound. And here the standard deviation is shown by the error bars. And we looked at any then that were above that standard deviation as potentially neuritogenic in this initial screening assay. And those are marked by the green asterisks here. We then wanted to confirm that we also saw similar changes in the complexity by looking at the number of processes coming directly off the soma and the total number of branching by these various compounds. And consistent with what we saw in the last slide, we saw many of those same compounds were again indicated based on the number of processes and the number of branching. So from there, we unblinded those compounds and identified manzamine A, 8 methoxymanzamine A, 12 propoxymanzamine A, 12 isobeta-oxymanzamine A, ATL297, irisin 1, and palineurin. And we also have the veratridine positive control shown here in the bottom right. And that was our positive control voltage-gated sodium channel activator. So from there, we are currently doing dose response curves for the neurite outgrowth assay for those compounds that were identified as potentially neuritogenic. We're also going to use propidium iodide and fluorescein diacetate to do live dead assays assessing for neurotoxicity on a dose response curve for those that we're interested in as potentially neuritogenic, because we want to know at which concentrations they may also be toxic. But we also are interested in the ones that are potentially neurotoxic. As I mentioned before, both Prialt and tetrodotoxin are ones that are neurotoxic, but at lower doses have been shown to potentially be therapeutic in pain. These may also have other potential therapeutic properties as well. And then from there, we're going to further analyze potential pathways that contribute to neuritogenesis. And so now I'm going to talk about a compound that we have looked a little bit further into with some previous work that I've done to show how that pathway analysis goes. So One of the compounds that we've been very interested in is brevetoxin 2. It's a member of the brevetoxin 1 through 10 family that comes from Carina brevis, or the red tide. And it is a voltage-gated sodium channel activator. And so with that voltage-gated sodium channel activator on the presynaptic neuron, that can cause a release of glutamate. It can also, on the postsynaptic neuron, cause voltage-gated sodium channels to activate and causing an influx of calcium. But with that glutamate, it can bind to your NMDA receptor here, marked as GluN2B, which causes an influx of calcium. That influx of calcium then helps activate the light blue molecule CAMK2, which then activates the small G protein rack. And that small G protein rack becomes activated and activates downstream PAC1. And PAC1 then goes on to activate the cofilin cycle, which is important for the F-actin growth that results in that neurite outgrowth. Now, from these initial cell studies, we can then go ahead and take it into a mouse model. And so in this study here, we've used the epicortical brevetoxin, and we applied it in a post-stroke model because right now there is no therapeutics for post-stroke recovery beyond the initial TPA and other things. It's all physical therapy. Now, in that penumbra region, about 5 days post-stroke, you're going to have some cells that are amenable to that neurite outgrowth and connectivity, but they have a tonic GABAergic inhibition that's kind of blocking that activity. So what we did first is we did a slight food restriction, and we tested to see if the mice were left or right-handed. We then did motor training with whichever hand they were to have them reach out and learn how to eat various pieces of pasta. And then as day 0 marked here with the lightning bolt, we did the photothrombotic stroke, 1.5 millimeters from bregma, to affect wrist rotation and the strength in their three middle digits. Then we did a motor assessment the next day. At 5 days post-stroke, where we're in that tonic GABAergic inhibition, we drove the voltage-gated sodium channel activity by brevetoxin application. And then the next day, we assessed their motor activities. And at 10 picomole, 100 picomole, and 1,000 picomolar dose, we did see that they had increased ability to eat their pasta. They had increased or decreased foot faults, and their performance on the catwalk was unchanged, suggesting that improved motor assessment occurred at that 10, 100, and 1,000 picomolar doses. We also then looked at the histology, and we found enhanced neurite outgrowth and connectivity, suggesting that this could potentially be a novel therapeutic option for the treatment of post-stroke. And finally, I'm lucky to work with a very wonderful group of national and international advisors, staff, and students that help to maintain the global marine pharmaceuticals pipeline, where we track clinical and preclinical pipeline applications of various marine natural product drugs. We also highlight some important new papers on Twitter and Facebook, so please do check those out, as are located in the yellow box at the bottom right corner. And finally, I would like to acknowledge some members of my laboratory that are very important, including Jihad Aburis, who's my technician, Miriam Butt, an M3 medical student, Sujin Seo, an M2 medical student, and Jacob Draves, a master student here at Midwestern, who have all contributed to the Manzamine project work that we have going on. I'd like to thank our entire marine-pharmacology.org team. It's a wonderful team to learn about these various drugs. I'd like to thank the Midwestern University Department of Pharmacology for supporting my work. My collaborators, including Dr. Alejandro Mayer here at Midwestern University, Bill Gerwick at the University of California, San Diego, Mark Heyman at the Medical University of South Carolina, and most importantly, my postdoctoral mentor and continued collaborator, Dr. Thomas Murray at Creighton University. Thank you for your time.